Hello, my name is Dan Tempest, and today I'm going to be talking to you about glycolysis, gluconeogenesis, and the role of aldolase in that whole process, including its expression. So, just to introduce you to the concepts, what are we really going to look at today? We need to understand the glycolysis and gluconeogenesis pathways. In order to do that, we will also compare them and look at some compare and contrast similarities and differences between them. Number two, how is the enzyme aldolase involved in both? Aldolase is a really key enzyme that is involved in both processes, plays a really key role. So we're going to look at that specifically and what's its function in the whole process. Then we're also going to look at the three different isoforms of aldolase, A, B, and C, and look at where they're expressed and their different expression patterns and what kind of some differences between them. And then the last thing we'll do is look at aldolase B expression in particular, and we'll look at 4 different types of transcription factors, as you see there, and seeing how they are going to impact gene expression of aldolase B. So here we go. So the first thing I need to go through is glycolysis, which is a 10-step reaction, it's 10 steps, and this is an anaerobic process, which means it does not require oxygen. This is the first step of cellular respiration. So depending on if there's oxygen present or not, will impact essentially where this pyruvate ends up. If it ends up going into the mitochondria, there would be oxygen present, and it would be an air ant that what follows with the Krebs cycle or citric acid cycle, and then oxidative phosphorylation would mean there's oxygen present. And if it doesn't, it would move into fermentation, which would allow the NADH to be remade into NAD plus so that glycolysis could continue. There's really 2 key phases of glycolysis. Let me move this out of my way here. And the first one is on the left here, our energy investment phase. It's known as the energy investment phase because of 2 key parts. One is right here, where we use an ATP, and the other is again here, where we use another ATP. So it's the energy investment phase because ATP is being used, and it's 2 ATP that are being used. So in that first step, we have glucose. Glycolysis literally means the splitting of sugars. So glucose is generally that sugar. Glucose is broken through using the enzyme hexokinase into glucose 6-phosphate. That's our first step. The second step is glucose 6-phosphate using the enzyme phosphoglucoisomerase is made into fructose 6-phosphate. Okay, and then in step 3, again, we use an ATP here. We're going to use phosphofructokinase, which is a really key enzyme in this process as it regulates the rate of glycolysis and whether it happens or not, essentially. Really key enzyme here is phosphofructokinase, and that enzyme makes fructose 6-phosphate into fructose 1,6-biphosphate. So again, that ATP added another phosphate there, and then the other phosphate was added here by that first ATP, and that at this point, that one is right there. So here is where we get introduced to aldolase. Aldolase, again, a key enzyme in the process. And aldolase's job is to break that fructose 1,6-biphosphate in half, essentially. So we're going to lysis break it. So we're going to have 1 dihydroxide acetone phosphate and 1 G3P, which is glyceraldehyde 3-phosphate. And those can really be interchangeable, and they're interchangeable using the enzyme isomerase. Okay, so that completes our energy investment phase and leads us to the second part of glycolysis, which is our energy payoff phase. In the energy payoff phase, we get to harvest some energy, and that energy comes in the form of 4 ATP. We see that here, 2 plus 2. However, most of the time we like to tell students it's that glycolysis makes a net of 2 ATP. Net of 2 because we create 4 in the energy payoff phase, but we used the 2 in the energy investments phase, so 4 minus 2 would equal 4 or 2 net. The other piece of energy that we do create is 2 NADH, and these 2 NADH can then be transferred into the mitochondria to be utilized in oxidative phosphorylation, where they would drop off their high energy electrons at the electron transport chain. or they could be used by fermentation. And depending on the type of fermentation, if you're doing an alcohol or lactic acid fermentation, would depend on the final electron acceptor there. If it's an oxidative phosphorylation, the final electron acceptor would be oxygen. If it's alcohol fermentation, ethanol would be your final electron acceptor. And lactate in lactic acid fermentation would be your final electron acceptor in that form. But just to go through the remaining 5 steps here, in step 6, again, we have those G3P in the dihydroxyacetone phosphate entering here. And this would turn that in, step 6 would turn that into 1,3-biphosphoglycerate. Then in step 7, and in step 6, just to reiterate, that's where you would make those 2 NADH that are a product of glycolysis. In step 7, phosphoglycerylkinase would create 2 ATP and make that 1,3-biphosphoglycerate into 3-phosphoglycerate. One thing that is a key thing to point out, now we only have that 1 phosphate left because those 2 ATP were created using the other phosphates. Now, another thing that I do want to note on this energy payoff is on this upper left-hand corner of each white box, it indicates how many of these molecules we have. So each one of these has 2 of these molecules going through that process. And that's, again, because aldolase broke that fructose 1,6-biphosphate into 2 molecules. So that's that reason why we need to note that there are 2 molecules moving through this process. In step 8, phosphoglyceromutase would turn that 3-phosphoglycerate into 2-phosphoglycerate. And then in step 9, enolase would remove some water and create phosphenolpyruvate. And then in that last step, pyruvate kinase creates 2 more ATP in the formation of 2-pyruvate, which is another key product of glycolysis. As mentioned, that pyruvate, where it ends up, really determines what determines that is oxygen is present or not. And under aerobic conditions, that would enter the citric acid cycle. Really, the link reaction would turn that into acetyl-CoA, which would enter the citric acid cycle. So that is glycolysis. And we need to compare that to gluconeogenesis. Now, what I first want to point out is this left-hand picture is why we really need gluconeogenesis. So our brain uses a lot of glucose each day. And half of that glucose would come from storage of glycogen that is found in the liver. However, if you go through a fasting period or if you're going through some rigorous activity, you might run out of that supply. And gluconeogenesis essentially allows us to raise our blood glucose levels through kind of a reversal. So you'll notice over here we have glycolysis, and that's happening here in the skeletal muscle. But here we have our gluconeogenesis over on this side. And the big idea here is we're able to create more glucose, which would raise our blood glucose levels. There's 3 carbon compounds such as lactate, such as also pyruvate and glycerol, as well as some amino acids can do this. So this could be, again, here we have lactate down here. Here's pyruvate. But again, that's not quite limited to only those. But we're making new sugars here. I want to point out that the reaction of gluconeogenesis are regulated by insulin, glucagon, and cortisol. Each one of those help to regulate this process if it's needed, if it's not, how fast it happens, etc. And then I do want to point out as well, because when I get into this right picture, it kind of some people could get a misconception that these are just running in the opposite direction, and they're essentially interchangeable concepts. And they're not. 7 of the 10 reactions are interchangeable. I'll show you some of those, but there are 3 reactions that do make them independent of one another. So it's not necessarily a perfect a reversal of the two. So I want to point that out. All right, so a couple key things to point out here. Again, on the left side, I have glycolysis, which I went through on the previous slide. But now we have this gluconeogenesis on the right side. And it's in the blue. I want to point out a key comparison as this presentation focuses a little bit on aldolase. I want to point that out. So that is right here toward the top third. Again, that's where that fructose 1,6-biphosphate would get split. But aldolase is one of those 7 enzymes that can go in either direction. So you'll notice on the left, the glycolysis reaction is moving in that direction, whereas the gluconeogenesis, it moves in the opposite direction. Key thing here again, it's a way for the body to create new glucose. So in gluconeogenesis, we're going to synthesize glucose to raise blood sugars during times of fasting or rigorous exercise when we need to raise that blood glucose levels. Okay. Clear the screen here quick. So here's aldolase again. Its key role. Aldolase's job is to break this bond here. And in doing so, it breaks that fructose 1,6-biphosphate. Again, that's on the left side of our reaction here. Uses aldolase. Aldolase is that enzyme that's catalyzing this reaction. And we break it into dihydroxyacetone phosphate and G3P, which is glyceraldehyde 3-phosphate. And we are able to make a little over almost 24 kilojoules per mole of energy. This is Gibbs free energy here. So amount of energy the cell essentially can harvest from this reaction. Okay, so this is aldolase's function. Again, I want to point out this arrow goes in both directions. So if we're going in this direction, breaking that would be in glycolysis. And if we're going in the opposite direction, that would be the gluconeogenesis. Making potentially glucose in that reaction. So aldolase functions in both directions. All right, so now we're going to look at 3 isoforms of aldolase. So this image here on the right does a nice job of kind of showing each one's role, and they're labeled right here. So again, we have that fructose 1,6-biphosphate right here, and that can be broken down by aldolase. It can also go in the opposite direction during gluconeogenesis. This does a nice job at showing the difference, though, in fructose metabolism up here versus glycolysis. So it can break down glucose. It can also break down fructose in this process. But aldolase A isoform, it's predominantly located in muscular tissue. Can be found in other tissues as well. It's also very active during the embryonic development, and it is actively involved in glycolysis. So you can see here on the right side of this image over here, we have aldolase A being actively involved in glycolysis. It also has a higher affinity for fructose 1,6-diphosphate than aldolase B does. Just important to point out a key distinguisher between A and B. Okay, so that's aldolase A. Again, key thing, it's expressed in the muscular tissue, also can be found, and it's also very involved with the embryonic development. Okay. So now moving into aldolase B isoform. Again, I'll point out aldolase B is on both sides of this diagram. It's actually the only one that is involved with both sides. Key expression, it's predominantly expressed in the kidney, but also in the liver and the small intestines. It is also not required for gluconeogenesis like A and C can be. Okay, so this one is not required. So I do want to point out that it is a key factor in fructose metabolism, though, as it is the only one located on that left side. So that's aldolase B. We'll look at more on aldolase B and how its gene expression can be impacted by activators and inhibitors here in a little bit. Our last aldolase isoform is C, and C is predominantly expressed in the brain, As I mentioned earlier, the brain requires a lot of glucose. So this is going to be a key isoform in helping get the brain glucose. So C again is only located on the right side of this image here. We see it right there. So it's involved with glycolysis. As I mentioned, predominantly expressed in the brain. It also has a higher affinity for fructose 1,6-diphosphate than aldolase B does. So similar to aldolase A in that manner, but different in where it's expressed. This is mostly in the brain, as I mentioned. All right. So that culminates our look at the three different aldolases. Now we're going to look specifically at aldolase B and its expression and the transcription factors that are involved. So there's three different activators that would stimulate gene expression of aldolase B. So I want to kind of couple these three up here. DBP is an activator, HNF1 is also an activator, and C/EPB. Those three are activators. Again, an activator essentially is going to stimulate or encourage gene expression to happen of aldolase B. So aldolase B is the enzyme, the protein that would be formed as a result of gene expression, and these three are all going to help that to happen. On the other hand, HNF3 decreases expression. It does that by acting as an inhibitor. And there's two really key things that it does as an inhibitor. It's a competitive inhibitor, so it's competing for the active site of HNF1. So they kind of antagonize each other. They compete for that same active site. So HNF1 activates, HNF3 deactivates or inhibits this process. And the other way it acts as an inhibitor is it's an allosteric inhibitor, so non-competitive now. We're not competing for the active site. We're going to bond somewhere else other than that active site and still stop DBP. It's an allosteric inhibitor of DBP, that other activator that we mentioned up above. So these are, again, four different transcription factors that are going to impact, does gene expression happen? Do we create aldolase B or do we not? Do we want to inhibit that? So some key distinguishers between those four, sorry. All right, so just to reiterate, again, what have we learned today? We've talked about understanding glycolysis. We went through that 10-step anaerobic reaction, first step in cellular respiration. Breaking the glucose down into 2-pyruvate and the 10 steps that involve that, all the enzymes that are involved with that. We create 4 ATP, but it's only 2 net because we utilize 2 in the energy investment phase. We looked at those energy investment and energy payoff phase. We also compared that to gluconeogenesis. We said that they're not perfect opposites, although that diagram does kind of create that misconception a little bit. Gluconeogenesis creates glucose. Could be from lactase, could be from pyruvate. It can also be from glycerol. So gluconeogenesis, the big idea is it allows during a time of fasting, during a time of rigorous activity where we need to raise blood glucose levels, gluconeogenesis allows us to do that. We also looked at how the enzyme aldolase is involved. And remember, aldolase's key role is to take that fructose 1,6-biphosphate, break it down into potentially G3P, where that's a key breaking point. So glycolysis, lysis means to break. But aldolase was also that enzyme that could take those 2 pieces and make them into the 1 during gluconeogenesis. So it plays a really key role in both processes. We also looked at 3 different isoforms, A, B, and C, and we looked at where they were expressed. So this image again on the right here does a nice job of showing where each one is. So if we look at aldolase A, place where that one was expressed here was in muscles. It's also found in some other tissues and very key in embryonic development. If we looked at B, it was involved with the kidneys, the liver, and the small intestine. So we show that here. And then C was a key spot in the brain. So just distinguishing those 3 does a nice job of showing that. We also looked at some key differences A and C are very involved with glycolysis, where B is very involved with fructose in its breakdown. And then we looked at aldolase B in particular, and we looked at its gene expression, specifically how transcription factors C slash EPB, DBP, and HNF1 are all activators. So if I separate those three, and then HNF3 was an inhibitor, and it did that through competitive and also allosteric means. So I hope you enjoyed, and I hope you learned a lot about aldolase, gluconeogenesis, and glycolysis, and kind of how they're all related and how they can be impacted. I do want to show you quickly my references. Let me clear out my writing here, and I hope you enjoy your day. Take care.